Hello and welcome to our next lecture. This is cell cycle and mitosis. So before watching this video, I highly encourage you to review the lecture slides for components of cells. I didn't make a lecture video specifically for that topic. It's pretty self-explanatory. If you go through the slides, you can read about the different components of cells, specifically paying attention to things like the nucleus and what's in the nucleus. Also, just sort of briefly going over some of the systems in the cell that help transport materials in and out. Mostly, however, what we're going to focus on is our understanding of the genetic information inside of the nucleus, which of course is DNA. So again, review that slide, those lecture slides first before you actually watch this video. That's my recommendation. And then I think this particular lecture will make more sense when you've done that. All right, so let's get started. So what we're going to be looking at in this particular lecture is we're going to talk about the life cycle of a cell. We can kind of split the life cycle up into two parts, one part being interphase, and the next part being mitosis, which we use PMAT to represent the different phases of mitosis. So let's start with the cell cycle. You can think of the average cell spending most of its life in this green portion right here. This is referred to as interphase. And this is when the cell is doing its normal cellular functions. So a heart cell is behaving like a heart cell, and a muscle cell is behaving like a muscle cell, and a liver cell is behaving like a liver cell. So whatever it is their specific function is what they're mostly doing. However, as they are doing these functions, they are also growing. And when the cell becomes of a particular size, it can then divide and create a whole new cell. And that's this part right here, which we call the mitotic phase, which we're going to go into more detail on that. So there are very important steps that have to happen before a cell is ready to divide. And I mentioned one of them happens to be the size of the cell. It needs to grow to an appropriate size to be able to split into two. But the other thing that we need to do is we need to make an exact copy of all of the genetic information that's inside that cell and then pass that along to the new cell that we're going to create. So when you were looking at the components of cells, and we talked about DNA, remember, is split up into pieces. We call those chromosomes. But when the cell is doing its normal cellular duties, the DNA itself is loose and sort of unwound, and it's like spaghetti is kind of the best way that I could represent what it looks like. That's important for it to be loose and unbound because it makes it easy to access the genetic information. Remember, the genes in our DNA code for specific proteins, and then those proteins are needed for these cells to carry out their functions. If the DNA is tightly wound up, you can't access those genes. So when the DNA is in interphase, all that DNA is just loose. Before we can go through the process of cell division, one of the things we need to do is we need to create these short condensed chromosomes that are going to be easier for us to essentially move around during this mitotic process. So we also then will again make a copy of all of those chromosomes. We copy all the chromosomes, we make them short and fat and easy to move. And the idea here is that we are making a copy of every chromosome that's inside that cell. And we're going to refer to these copies as sister chromatids. So sister chromatids are, again, an identical genetic copy of the information that we need to pass along to the new daughter cell. So here's what it looks like. Again, here's an unreplicated chromosome, right? It's kind of loose, looks like loose yarn or spaghetti or whatever analogy you'd like to use. This is great because we can easily access the information, but when we want to actually move these chromosomes around, it's not easy to move a loose pile of spaghetti or a loose pile of yarn. What we want is something that is concrete and something that can easily be divided. So here's the same chromosome. Now it's right here, and we replicated it. We made a copy of it. And it's attached at a point called a centromere. So you can also see the centromere right here. That's just the point of attachment for those two sister chromatids. We'll talk about more of that later. So mitosis is a type of what we're calling an asexual division. Asexual simply means that there is one cell and one cell only that's involved. We are not introducing new genetic material as we would in a type of sexual division, which we'll talk about in the next lecture when we talk about meiosis. And this is, again, a part of the cell cycle. So it's a normal occurrence in cells. The goal of mitosis is to make cells that are exactly the same as the original cell. That heart cell, we're going to make a copy of that heart cell, and it's going to do the exact same thing that the previous heart cell did. So there are 3 steps, once again, in what we're referring to as the cell cycle. I'll go over them briefly each. Interphase, mitosis, and then a step called cytokinesis. So if we looked at sort of the mitotic portion of this division, we're starting with our original cell. We're going to, during interphase, we're going to make copies of all of that. Then we're going to start to move these things around. We will have a process through which mitosis, we split things up, and then we're eventually going to end up with 2 copies that look identical to the first copy we started with. So these are referred to as daughter cells, and then the original cell is usually referred to as a parent cell. So interphase. Interphase, again, is the normal life of a cell. This is what a cell is mostly doing. We can also separate this out into stages. So we call it the 2 gap phases, gap 1 and gap 2. And then S is the synthesis phase. Again, your gap phases are, oops, I'm sorry, I moved that up too quickly. Let's go back up. Oh, well, now hold on. Where's my page up? Sorry, apologies. I'm using aha, there we go. I'm using a laptop that I don't normally use to record this lecture. So it took me a second to find the page up button. So in any case, again, your gap phases are essentially what you can think of as the phases where the cell is growing. And we are making sure that the cell is big enough. The synthesis phase is where we are replicating the DNA, right? So we are making copies of all of those chromosomes so that we are able to condense them and move them around. Okay, so again, gap phase, it's growth. We make copies of all of the organelles. So again, from the previous lecture slides, you know what all the organelles are. The cell gets larger, it gets big enough to divide. Synthesis, we're going to make copies of all of those chromosomes and get them connected by centromeres so that they're ready to move. And then in the second gap phase, we're making all the proteins that we need for mitosis. So just like everything, proteins are sort of the jack of all trades when it comes to cells. They do a little bit of everything. And in this case, we need some proteins to help us get this mitotic phase started. So the purpose of mitosis is to separate the sister chromatids that we created during the synthesis phase of interphase and put them into 2 different cells so that each cell has a complete set of chromosomes. We can break mitosis down into prophase, metaphase, anaphase, and telophase, or PMAT. That's where we got PMAT. So let's talk about the first part of mitosis. In prophase, we see the replicated chromosomes condense, right? So we have all of this loose genetic information that's really hard to move around. So we're going to make them short and fat and easy to move. We also create special structures called microtubules. Those microtubules are going to form at the poles of the cell. Poles is a fancy way to say the ends of the cell. So just like the North Pole and the South Pole of the planet, you can think of the North Pole and the South Pole of the cell as well. Nuclear envelope disintegrates. Okay, so remember we had a nucleus that contained an envelope with pores in it that small things can move in and out of, like RNA. Can't have giant chromosomes moving around if they're stuck inside the nucleus. So we're going to disintegrate that. Next comes metaphase. In metaphase, we take all of those replicated chromosomes and we line them up across the equator of this cell. Equator of the cell is the exact same meaning as the equator of the planet. Okay, so same idea, middle of the cell. Those microtubules that are formed at the poles of the cell, they're going to reach out and attach to the centromeres of those sister chromatids. All right, so let's take a look. Here's prophase. So in prophase, the genetic information, the chromosomes become visible. If you look at a cell that's in interphase, you can't see this. This isn't visible. So It's visible now because we have condensed those chromosomes and we can actually begin to see them. The nuclear envelope also has disappeared, okay? And now we're ready to move into metaphase. So in metaphase, we have taken those chromosomes and we have lined them up here at the equator of the cell, kind of the middle of the cell. We also have those microtubules, which are reaching out from either direction, and they are latching on to the centromeres of our sister chromatids. Next is anaphase. In anaphase, we're going to split the centromeres. Those microtubules are going to contract, and they're going to pull those sister chromatids apart towards the poles. So they were all lined up in metaphase. Now we're going to pull them apart in anaphase. Telophase is the last part of mitosis. We're going to condense, we're going to recreate the nuclear envelope around those chromosomes. And the chromosomes are going to go back to their uncondensed form. They're going to go back to their loose spaghetti form so that they can actually enter back into interphase, and we can access those genes again like we needed to. Here's anaphase. In anaphase, you can see that the chromatids are being pulled apart. Okay, so those microtubules. The centrioles are just the point of attachment for the microtubules at the poles of the cell, by the way. Next comes telophase. So in telophase, you can begin to see two very distinct cells now, right? We have a group of chromosomes here. We have a group of chromosomes here. They are unreplicated, right? And we are beginning to go back to that uncondensed form. Now, even though the process is complete of separating the sister chromatids, we don't have two distinct cells yet. For that, we need to enter into cytokinesis. So cytokinesis is basically where we're cleaving that one giant cell into two cells. So we've got an equal distribution of genetic information. We have an equal distribution of organelles and the other molecules we need for the cells to function. Now all we have to do is actually split that cell into two. We use some microfilaments that we will spin around the furrowing point or the point where the cells came together, and then we'll literally just pinch it in two. That's all cytokinesis is. All right, so once that occurs, we have two daughter cells, and those two daughter cells then go right back into interphase, and they begin the whole process all over again. The cell cycle rate varies in different tissues at different times. So there are certain tissues in your body that need to generate cells more quickly than others. Your skin is a great example of a tissue that has a very high turnover rate. You are constantly sloughing off dead skin cells. So your skin cells will replicate much more often than, let's say, your heart cells do. And these are all internally controlled by something called a telomeres, if you want to think of it. It's kind of like in a turtle clock. Telomeres are these little protective ends on the ends of the chromosomes in these particular cells, and depending on the length of the chromosome, or excuse me, the telomere, depends on how many times these cells are capable of dividing. Every time they divide, the telomere will get shorter and shorter and shorter until it's too short. You can think of telomeres kind of like a protective cap on those chromosomes. And once that protective cap is too small, we can't reliably make copies anymore because we'll probably make too many errors. And as you'll see later in the semester, errors in DNA replication can be bad. All right, so what's controlling cell cycle? So how do we know when to move from GAP1 to synthesis to GAP2 to mitosis? There's a whole host of checkpoints that are involved. Some of these are internal and some of these are external. And what's really interesting to note is that cancer, which is a form of uncontrolled cell growth, so this is when your body is not in control of the cells that are replicating over and over and over again and creating a mass, which we call a tumor, for example. Those result from faulty checkpoints. So when something happens and something's wrong and the cell cycle goes out of control, then we begin to see different types of diseases. So one checkpoint is here. This is a growth checkpoint. Is it big enough? Do we have all of the proper proteins? If not, it's going to go through the gap again. If it is, we can move on to the synthesis checkpoint. It checks whether the DNA has been replicated correctly. If so, we can continue then on to the second gap and mitosis. And mitosis only occurs if we have the chromosomes that are ready to go. And once the chromosomes are ready to go, we can go ahead and enter that cycle. A mistake in any one of these checkpoints, and there are genes that are responsible for checking all of this, a mistake in any one of these could potentially lead to uncontrolled cell growth. A little bit about telomeres here. They're located at the end of your chromosomes. Like I said, you can think of them like a protective cap. It's a 6-based DNA sequence that can contain anywhere from hundreds to thousands of repeats. Most cells will lose about 50 to 200 of those telomere bases after each cell division. And a typical cell can go through about 50 divisions before it's too short and it can't divide anymore. There are certain cells, sperm, egg, bone marrow, and cancer cells, produce something called telomerase, which actually prevents the shortening of telomeres. So you can think of it as kind of a way to make sure that you can produce those cells over and over and over and over again, versus what you see in other cells occurring, which is basically a lifespan, right? You can only divide so many times before you're no longer able to anymore. All right, so at the end of these lectures, as I mentioned, I think in the Intro to Genetics lecture that you saw, this is the lecture review slide. These are the important terms and concepts that you are responsible for knowing after watching this lecture.